guys. Just to get your attention on some really exciting things coming up with MedSoc. So the SB Downton Leadership Day thing is coming up on the 11th of August, which is a Saturday. It starts at 10 a.m. and it'll be a really good opportunity to think about future medical practice as well as other leadership things. So check that out on Facebook and on the UNSW MedSoc page. And the other thing is integration party is next Thursday night, so save the date. We'll have another video coming up soon. And yeah, get excited about that.
Oh, hi guys. So hope you've been checking your Facebook. So the next set of OSCE sessions is today. It's from 11 a.m. to 1 p.m. It's College C. But even if you're from another college, if you can't make it for the other OSCE sessions, you're free to come. This is on the respiratory session, and because you're in College C, I think you have SGS today, and you're meant to bring a stethoscope, so bring a stethoscope for that as well. And yeah, so it's in SG rooms in Matthews. Thanks.
Okay, we'll get started again. So we ended the last lecture with digestion and absorption, and now we're going to throw in a different component to that, or a new component. So we talked about digestion in the small intestine, absorption by these enterocytes lining the villi of the small intestine. What then happens inside the cell is we have transport to the endoplasmic reticulum, and the endoplasmic reticulum, as you'll probably be aware in terms of what different organelles do in the cell, does a lot of synthesis, including synthesis of lipids. So that's where the triglyceride synthesis is occurring. And if you think about it, we've just broken down triglyceride to get into the enterocytes. Now we're building it up again. Does that make sense? That's just the way it works. Probably because triglycerides in bulk would be relatively difficult to get across into the enterocytes fast enough. What we then have is we have formation of a lipoprotein. So a lipoprotein just means lipid and protein, and so it's basically a way, as we'll see, of transporting lipid around the bloodstream. The chylomicron goes via the lymphatic system and then into the blood system. So that's what happens with regular fat, and what I'm talking about is most of the fat that we would be consuming. Carbon chains of fatty acids would be at least 16 carbons in length, maybe 16 to 20 2 carbons in length. And whereas for shorter chain or medium chain fatty acids, it doesn't need to go through this palaver, and it can actually go straight to the liver via the portal system. And why would that make sense? At least for some individuals. It again comes back to the milk drinking infant. So remember, the milk drinking infant, because the triglycerides that they're consuming in the milk is more medium chain, that you have a couple of extra lipases which can come into play. Lingual lipase in the mouth, gastric lipase in the stomach. And here we've got another way of saying, well, okay, let's just get that fatty acid immediately to the liver, where it can then be redistributed for energy or for building. And this diagram basically shows the same thing. So small lipids, C4 to C12, can go straight to the liver via the portal system. Large lipids greater than C12, that's 12 carbons in length, need to be packaged into these chylomicrons, these relatively large particles, which then go via the lymphatic system into the blood circulation. Okay, so what we're going to try to do here is build up, through this lecture and the next lecture, how cholesterol can contribute to heart disease. And what we're going to be dealing with today is how the cholesterol is transported around the bloodstream. So you've had lectures on atherosclerosis, presumably, maybe second years have they? Really? Wow. Okay, so it's major killer, obviously, in Australia as in other countries in the world. Like a lot of these diseases, they're becoming much more prevalent in China, Southeast Asia, India. So they were always seen to be this sort of considered to be associated with affluence. Obviously, affluence is growing. In Australia, there's often a correlation with deprivation, actually, with heart disease and with other more developed countries. So atherosclerosis, basically, as we'll see in the next lecture, a lot of the gunk, which is... does anybody know what atherosclerosis means? So sclerosis means hardening. Athero comes from a Greek word for a weak porridge-like substance. And that's why we use Greek or Latin terminology in medicine, because it sounds a lot more serious if you're saying to a patient, I think you've got atherosclerosis, rather than you've got porridge hardening. You know, it just it doesn't have the same gravitas. And there are many risk factors which have been pinned down on heart disease. At last count, there's probably 232 risk factors for heart disease, and this is one of them, inverse association between sense of humour and coronary heart disease. And I was very disappointed because they didn't actually say what jokes that they used to define this. But the 8 classical risk factors for heart disease are these. Age, sex, obviously men have much greater risks than premenopausal women, family history, and so these are things which we can't do too much about. So they're considered non-modifiable unless you go in for some expensive surgery. And underpinning some of these modifiable ones, it's genetics as well. Obviously there's always gene-environment interactions. And that's certainly the case for things like cholesterol and blood pressure. There's relatively strong genetic components there, polygenetic usually. Smoking obviously is something that can be done something about, and diabetes mellitus is also considered to be a risk factor for heart disease, as well as lack of exercise. And so it all comes down to that sort of metabolic syndrome. Obesity is linked into it as well, obviously. So as those sort of lifestyle diseases, obesity and diabetes increase, we would then expect more heart disease as well. So in the 1960s, we reached the pinnacle of this coronary epidemic in Australia, and its rates have been going down since, but they're actually predicting that they're going to start going up again because of increasing obesity and diabetes. So it's worthwhile pointing out that these only explain 50% of the incidence of cardiovascular disease. And it's worthwhile noting that there are other risk factors which are now being used much more clinically, such as C-reactive protein, which is a measure of inflammation. Heart disease really is chronic low-grade inflammation. That's how it's coming about. But because this is a cholesterol lecture, I'll be focusing upon the cholesterol aspect here. And so this might be something that you might have a variant of this slide in your notes. Don't worry too much about it. All it's doing is showing where some of the epidemiology came from. So the Framingham Heart Study, have you heard of Framingham? Small town in Massachusetts, where they've been studying them since 1948. And they made some of the first links between cholesterol levels and heart disease. And this is from a huge meta-analysis of 61 prospective studies representing 900,000, close to 1 million participants in all of these 61 studies. And what it shows you is a very robust relationship between increasing plasma cholesterol, increasing heart disease risk across age groups. So every age group, you've still got a relationship there. And I want to point out that cholesterol, there's more to heart disease than cholesterol. It's not the only thing, but again, obviously that's what we're focusing upon in this lecture. And the reason that we need to understand a little bit about lipoproteins and lipoprotein metabolism is that this blood cholesterol measure is really just a snapshot of a very dynamic process. So you get a measure out, 5.2 millimole total cholesterol, and you try to work out, well, what does that mean? And you probably need a bit more information, and you need to know, what that cholesterol might be doing. So first off, we're going to just, very simple, we're just talking about the balance of cholesterol in the body. And so we have input and output. Input is cholesterol that we absorb from the diet, as well as cholesterol that we synthesize. Output is mostly catabolism to bile acids. So we've learned in the previous lecture that cholesterol, it can be converted into bile acids. but we can also excrete some of that cholesterol into the bile and then into the faeces, and that's how it escapes. So just as a little aside here, the reason, so cholesterol is, from an evolutionary point of view, very important, and we have something like 100 different proteins which are responsible for making cholesterol, shuttling it around the body, and yet we get no energy from cholesterol. So that's telling us that cholesterol is important for the body. And so that's part of the problem with it. It's often worthwhile thinking about an evolutionary point of view for these things. So if you want to make a molecule of cholesterol, you need 10 times more energy than you need to make a molecule of glucose. So what that's telling you is that the chances are you're going to have mechanisms in place for hanging on to that cholesterol. And again, if you're hunting woolly mammoth, that's a good thing because it's expensive. It's expensive to get cholesterol from the diet because a saber-toothed tiger might get you, or from making it from the de novo, from scratch in the body. So, in other words, we've evolved mechanisms for hanging on to cholesterol, which is great when there's not enough, but when there's a lot of cholesterol, a lot of fat, it's not always such a good thing. So, just going back to that cholesterol homeostasis, I'll throw some numbers at you. These are very rubbery numbers. But, for example, you might be consuming, regular diet, you might be consuming about 400 milligrams per day of cholesterol. And this might come from various animal products, eggs, prawns, dairy products, meat. You've got a total cholesterol pool of about 100 grams. What you should note is that you might be synthesising 800 milligrams per day. So most people don't really think about that. We actually usually synthesise a lot more cholesterol than we take in the diet. And then we balance that by excreting bile acids and biliary cholesterol. So if you add those numbers up, you're actually balancing input and output. So I don't know if you've seen these avocados in your local fruit and veg shop. Occasionally you have these cholesterol-free sticker slapped onto it. Has anybody ever seen that? It's a little bit like putting an alcohol-free sticker on a litre of milk. It doesn't make a lot of sense because plants don't have cholesterol. Plants don't have cholesterol. They have another sort of sterol. So you've probably seen these margarines, Proactive, Logical and things like that. They have these plant sterols which can actually compete with cholesterol for absorption, which is the way that they can lower cholesterol a little bit. Eggs contain quite a bit of cholesterol. So would we agree with this? Hands up if you agree with this statement. Cholesterol-free means a product is heart healthy. Hands up if you don't agree with it. Okay, few hands going up for the... so maybe I don't need to explode this myth, but I will anyway. So this is like a Mythbusters, nutritional Mythbusters website. And this is the relevant statement here. Blood cholesterol is a contributor to heart disease. But what's more important is the quality of the fat, the fatty acids that you're consuming, whether they're saturated in trans fatty acids versus unsaturated fatty acids. Because they're the things which tend to increase your blood cholesterols much more than dietary cholesterol does. And so I think that's a common misconception. So, for example, people still avoid eggs, which, are pretty good in moderation, good form of nutrients, and yet because they hear that they're cholesterol rich, they should avoid them. It's just that often a lot of foods which contain a lot of saturated fat often contain a lot of cholesterol. So that tends to muddy the water somewhat. Okay, so we talked about that before. So what we're going to be talking about now is the lipoproteins. And so lipoproteins, lipid transporters, why do you think I talked a little bit about evolution. Why do you think we've evolved lipoproteins? And there was a key phrase in the last lecture. When we did our experiment, can you remember when we did the experiment with the water and the oil? and there was 2 layers. Oil and water don't mix. So just imagine that you need lipids. You know, it's a great form for storing energy. It's a great form for getting quick energy for certain tissues, such as heart muscle. So we need to be able to transport around the body. It's needed for building cells. So we need, rather than having just globules of fat just sort of floating around, sticking everywhere around the blood system, around the circulation, we need a way to actually control that lipid movement and to make it actually soluble. So we have lipoproteins to do that. So they make the lipid water soluble, and we'll see how they do that. So these are some of the principal classes of lipoproteins. So we have chylomicrons and chylomicron remnants. And you will notice that the chylomicrons are shown as 1,000 nanometer diameter. How many microns is that? 1. Yeah, which is why it's called a chylomicron. Then we have VLDL, IDL, LDL, HDL. And so we've got all of these sort of weird letters which basically allude to what density it is. And we'll talk about why we've got these in a minute. And so these guys are considered sort of the bad guys, and this is considered the good guy. And we'll come back to that in the next lecture, why that's the case. So first off, the general structure of a lipoprotein. And so the lipoprotein, as I hopefully mentioned, is a vehicle. It's a way of transporting something around. So we have a cargo inside, and the cargo is neutral lipids. And these include triglyceride, which we heard in the last lecture is neutral, as well as cholesterol esters, which are. On the surface, we have some proteins, which are called apoproteins, and there's a whole bunch of these. There's at least 20 of these, and we won't be talking about these to any extent at all. Just realise that there is a protein component, which is why they're called lipoproteins. And we have phospholipids, and phospholipids, remember, they're amphipathic, and so they can act as emulsifiers on the surface of the lipoprotein to help it be nice and soluble in the bloodstream. And then we can have free cholesterol on the side as well. So we'll talk about each of the major lipoproteins. And so first off, we're going to talk about those ones which come from the diet. And so because they come from the diet, they come from outside, exo. They're derived from outside, exogenous, which is what that means there. And so they transport mostly triglyceride. With the foods that we eat, what do you think the major lipid class is that we consume? So we talked about, for example, triglycerides, phospholipids, cholesterol, either free or esterified. What do you think is the major lipid class that we consume? Hands up who thinks triglycerides. Yeah, it's triglycerides. Yeah, so probably about more than 95% of the food that we eat, fat that we eat, is triglyceride. As I said, that's the most common way that animals and plants store fat, so that makes sense. I mentioned 400 milligrams per day of cholesterol, so that might be less than half a percent of that, and maybe you might have a couple of percent of phospholipids as well. So the major lipid component is dietary triglycerides. So the fat is actually in this really represents pretty much the last meal or so that you've eaten. Then we have chylomicron remnants, and that the remnant means that it's derived from chylomicrons. And we'll talk about how it's derived from chylomicrons by introducing another lipase. So we've had a couple of lipases. We've talked about lingual and gastric lipase as important for milk drinking infants' digestion. Pancreatic lipase is the way that we do most of our lipid digestion. This lipase is actually on the surface of the endothelium. So the endothelium, as you'll know, single layer of cells separating the tissue from the blood, and it's on the surface of that. And what it does, it actually can chomp away at some of the triglyceride in the core of the chylomicron to make a smaller particle. And that'll hopefully become clearer as we go along. Don't worry, you look puzzled. So this is lipemia, which just means the increase in lipids in blood after a fatty meal. And so you can actually take a blood sample from someone after they've eaten a fatty meal or when they're non-fasting and leave it in the fridge overnight, and you'll actually get this creamy layer on top. And that's the chylomicrons floating up. And so this is lipemic, non-lipemic plasma, this is lipemic. So you can actually see it's cloudy, which is the chylomicrons. And this is the same thing from a dog where it's actually been centrifuged up. So you can actually, if you have a fatty meal, within an hour you'll have these chylomicrons appearing. And I'm going to introduce you to the way that lipoproteins traditionally have been separated. And not that you'll ever have to do this, but hopefully it gives you an idea of where these names come from. So here you've got whole blood, which you would spin. You've probably all had an experience of being at one of those pathology clinics, having blood taken or something like that, and they always have a little centrifuge nearby. And so that would be the first spin. And this is 2000 G, which means that it's 2000 times the force of gravity for 20 minutes. And so you've then got plasma, if there is anticoagulants in the tube, with the packed cells. And obviously that's red blood cells, white cells, platelets. If you then take the plasma and you spin it harder this time, so it's now 100,000 G. It doesn't need to be a really long spin. You can actually spin up those chylomicrons. If you now take the bottom part, the infranatant, and you adjust the density of it, so you just add all this salt to it and you make it a higher density, and which then enables you to just layer very carefully saline on top, which is what we've got there. Now we're spinning it at 350,000 G. So here, a gram marble would be equivalent to a 350 kilogram boulder. And so you've done pracs. How many of you had to use centrifuges? You've always been told to balance your centrifuge. If you're using one of these ultra centrifuges, if you get it wrong, the centrifuge starts walking out the door, or probably worse, starts walking through several walls. And I'm sure on YouTube there are probably many, there's probably a whole stream of linked videos of ultra centrifuge accidents. When I was a student at Sydney Uni some years ago, there was a devastating one at chemistry where it did, ultra centrifuge hadn't been properly balanced and did actually take out 2 walls. That's why we don't do this for a prac. Not that we don't trust medical students. So with spinning this, we can actually now start seeing other lipoproteins coming up. And so we have very low density lipoprotein right up the top. Low density lipoprotein sort of towards the middle of the tube, and high density lipoprotein at the bottom. And so hopefully you can now appreciate why we've got this weird terminology for these lipoproteins, VLDL, LDL, HDL. And you can actually see, you should be able to see LDL as a nice gold band here. And that's the carotenoids. I talked about carotene, provitamin A, and because it's carried mostly in lipoprotein in the blood. So I spent some time in Scotland analysing lipoproteins in middle-aged Scottish men who smoke. And you've all heard about the Mediterranean diet as the diet that we should be striving towards, you know, monounsaturated fat, lots of fruit and vegetables. The antithesis of the Mediterranean diet is the traditional Scottish diet. So if it's not deep fried, it's just not worth it. And so Scotland is home of the deep fried Mars bar. And this was actually in the Lancet a few years ago, where they actually thought, oh, it must be a myth. But then these doctors went out looking for the deep fried Mars bar. We conclude that Scotland's deep fried Mars bar is not an urban myth. Encouragingly, we also did find some evidence of the penetrance of the Mediterranean diet into Scotland, albeit in the form of deep fried pizza. So when I was in Scotland isolating these lipoproteins, the first time I did it, I thought, wow, this is incredible. They don't have lipoproteins. I'm going to get a science paper out of this. It's going to make my reputation. It never did, because it was completely wrong. Because they had lipoproteins, they just didn't actually have any fruit and vegetables with carotenes, so you could actually see it. So I ended up using a lipophilic dye to actually see the lipoproteins. So we talked about the exogenous lipoproteins. Now we've got the endogenous lipoproteins. So these are made from the liver, or made by the liver. And VLDL is very much like chylomicrons, but chylomicrons taking fat from the gut where it's digested and absorbed. Here VLDL is taking mostly triglyceride from the liver. And so mostly it's containing triglyceride that we've synthesized or resynthesized in the liver. And then we have VLDL has its own remnant particle, which is called IDL, intermediate density lipoprotein. In most individuals, you don't have too much of this because it's very transient, because it's formed and then it's quickly converted into LDL, and we'll see that in a minute. But again, this is lipoprotein lipase acting upon VLDL, chomping away at the triglyceride in the core, making a smaller remnant particle. And so it's relatively cholesterol enriched. And then we have low density lipoprotein, which is the way that most of us carry most of our cholesterol in the bloodstream. And this is derived from IDL by yet another lipase, so not lipoprotein lipase, but a lipase called hepatic lipase. And then we have HDL, and HDL isn't directly made from these, but there's contributions, which I won't go into because it's a bit complicated. And what we'll see is that in the next lecture is HDL is involved in a process called reverse cholesterol transport. And there's a lot of interest at the moment in trying to increase HDL levels in the circulation. So, but I might talk a little bit more about that in tomorrow's lecture. So if we had to just look at the composition, the percent composition of these major lipoproteins, chylomicrons, VLDL, LDL and HDL, what we have here is the yellow is the protein at the top. And so you can see that the protein is increasing, the proportion of the protein is increasing as we increase density. Why is that? The proportion of the lipid subsequently is decreasing. So the lipid to protein ratio is decreasing. So what did we say about when we did our very simple experiment, water and oil, and you all knew they didn't mix, oil was on top, and you all said, almost in unison, it was like a choir, that it was because it was less dense. So the lipid is less dense than the protein. So as you're decreasing the lipid to protein ratio, you're increasing density. Can you sort of get that? So the less lipid you've got, as in HDL, you can see half of its protein, half of its lipid, the higher the density of the particle. And the other thing this is showing us is the type of lipid that predominates. So triglyceride predominates both in chylomicrons and VLDL. And then we're getting increasing amounts of cholesterol in LDL, so that's about half the particle, a bit more phospholipid in HDL. Now if we were to take your blood now, quickly ultracentrifuge it, and measure the cholesterol in these major lipoproteins, VLDL, LDL, HDL, this is probably the way it would sort of look, where men would be definitely carrying most of their cholesterol in LDL. Women, because premenopausal women tend to have quite high HDL levels, which is thought to be one reason why they're more protected against heart disease than men. That they tend to balance the particles that they're carrying cholesterol in between HDL and LDL mainly. And if this was, this would be assuming it's fasting. If it's non-fasting, it doesn't actually make too much difference for cholesterol. It would if it was triglyceride, because you'd have all those chylomicrons with all the triglyceride. But there isn't going to be too much cholesterol in the chylomicrons. So in non-fasted blood, chylomicrons contribute maybe only to about 5% of the total cholesterol. So you might whack out your cholesterol by 5%. So often if you go to a pathology clinic and they just want your cholesterol, maybe they don't even want your triglyceride, and they say, oh, you haven't been fasting, you have to come back another time when you've been fasting. For cholesterol, it's actually not going to make all that much difference. But for other lipids, it will. Okay, so this is really just summarising the exogenous and the endogenous pathways. So we've got the exogenous pathway where we've got lipid from the diet via the intestine, making these chylomicrons. With the endogenous, we've got the liver making this triglyceride-rich lipoprotein, VLDL. And both of these particles are whizzing around the circulation and being acted upon by lipoprotein lipase on the surface of blood vessels, on the endothelium. And this might actually be, for example, if it's during exercise or something like that, exercise actually increases your lipoprotein lipase activity. And so it's a way that it's a great way of reducing lipids as well. And the free fatty acids that are released during this lipolysis, so the triglyceride in the chylomicron or the VLDL being acted upon by lipoprotein lipase, it's releasing free fatty acids which can then be transported attached to albumin. And albumin you'll probably know as the major protein in serum or plasma. So this is one of those slides that if you showed this in lecturing school, you'd fail because it's too text rich, but it's probably useful for you to look over later. So it's pretty much what we've talked about already. And so that's chylomicrons, and then the same thing with VLDL. So it's really just sort of summarising some of the things that we've said in words, just to study later. I prefer pictures. And so here we've got somebody eating obviously a very fatty ice cream here, and chylomicrons being formed from the intestine, whizzing around the bloodstream, being acted upon by lipoprotein lipase to make these chylomicron remnants. And that lipoprotein lipase releasing free fatty acids which can go to body fat, adipose tissue, for storage, or it can go to muscle for instant energy. And as I said, or I might have mentioned, heart muscle loves fatty acids, can chomp through quite a bit of fatty acid. And so those chylomicrons are actually very rapidly cleared by the liver. So obviously they're being formed quickly and they're being cleared quickly. That's the idea. In certain diseases they're not. And then obviously that's increasing more lipid going to the liver. Eventually that's going to be packaged into the LDL. It's not quite sort of as simple as it goes from here to there. It's going to stay in the liver for some time, and with different physiological stimuli you're going to make more VLDL. And so the VLDL does the same thing, whizzes around the capillaries, acted upon by lipoprotein lipase, free fatty acids going to adipose tissue and muscle for use, can then be converted to its own remnant particle, IDL, intermediate density lipoprotein, and then IDL should be pretty rapidly acted upon by hepatic lipase, I think somebody once asked me, why isn't this not in the liver if it's called hepatic lipase? And it's, hepatic lipase is in a lot of places, including the liver. It's not just the liver. And forming low-density lipoprotein. And so that's the way that we're carrying most of our cholesterol. Then that can transport cholesterol to peripheral tissues where it's needed for building up the cell. So we need cholesterol for a number of reasons. Can anybody name a reason why we need cholesterol? Hands up if you... Cell membrane, yeah, essential component of cell membranes. Without it, our cells leak like sieves. As a precursor for bile acids, and maybe somebody said it already, I just haven't heard. Steroid hormones, yep. Excess LDL can actually be taken up back by the liver. Obviously, the liver is a central organ in all of this metabolism. It's a big organ, and it can be taken up by the liver. What would you... It's taken up by receptors on the liver. You all understand what receptors are? What do you think would happen if you had a defect in those receptors? What would happen to you? Would your cholesterol level... Hands up if you think your cholesterol level would be decreased, your blood cholesterol level would be decreased. Unchanged? Increased? Yeah, it'd be increased. And we'll see in tomorrow's lecture that there is a devastating disease which does exactly that. So one thing that you should be taking away from this, so why do we need lipoproteins? So I've given you the answer earlier on in the lecture. Transport lipid around. What is it about lipid which makes it important to actually have a transport vehicle? The fact that it's not water soluble, yeah? The fact that it's not water soluble, we just don't want it glugging up our circulation in an uncontrolled fashion. We want to be able to transport it around. Why is HDL considered the good, so-called good cholesterol? It's not actually cholesterol, it's a lipoprotein, but it carries cholesterol. Why is it considered the good cholesterol? Anybody know? What's that? It transports cholesterol to liver from where? From where? From the periphery, from other places. Yep. We'll see why that's important. And so we'll hear more about that in tomorrow's lecture. Let me just quickly close this down, otherwise, just in case somebody needs to come in. Yeah. Is it used for... How do you mean preparing information?
Preparing. So you have one theory, so after coding information
repairing. Not really, no. So cholesterol is important in signaling, which is obviously all about information, and it's involved in signaling because on the plasma membrane, the cholesterol isn't just sort of randomly distributed on the plasma membrane, but you have what are called lipid rafts. And lipid rafts are where a lot of signaling molecules, proteins, kinases, etc., can actually aggregate, and so it brings them together, brings different signaling molecules together. So there is a connection there, but it's not quite as simple as I think you might be proposing. Sure, yeah, yep. Okay, so trans fatty acids are a type of unsaturated fatty acid, but the double bond is in a trans configuration, and so I showed you what trans look like, so straight chain, whereas cis configuration, which is the way that fatty acids normally are, have a kink in them, which makes them more fluid.
That one unsaturated.
Yeah, but they're both unsaturated. Trans is, it's just the orientation of the double bond. Double unsaturated just means it has a double bond. So both trans and cis monounsaturated lipids, they're both unsaturated. Yep. Yeah, it's just a configuration of the double bond. Yep.
Quick question. It's a little bit confusing for me. First of all, can I say lipoprotein as a container
Yep.
for transporting the cholesterol, triglyceride inside the blood? So for this one, is that produced in the food?
No, that is produced in the intestine. So remember, we've digested the fat.
Yeah.
We've absorbed it into those enterocytes in the villi of the small intestine.
Yeah.
We've repackaged it into a lipoprotein. That lipoprotein is chylomicrons. That then goes from the lymphatic system into the bloodstream and is then going around the blood. And so you can see that lipemia, the chylomicrons actually floating at the
So it's
top of the...
produced actually inside the small intestine.
Yeah, so it's from the gut. That's from the gut, that's from the liver.
So first of all, you got some food, you got some fat in the intestine.
Yep.
So you absorb the fat, you produce the protein inside the intestine. So it packaged inside
just packaging. Yeah.
like a vesicle.
Yeah.
So for this one, it means after it enter into your circulation,
Yep.
and
It's coming into contact with the lining of the blood vessels,
Yep.
the endothelium, where you have the lipoprotein lipase. And it's interacting, and the lipoprotein lipase is starting to actually hydrolyze the triglyceride, breaking it down, making a smaller particle, releasing fatty acids.
okay, inside it.
So the lipoprotein lipase can get into the, you're calling chylomicron the container,
Okay.
and actually chomp away at some of that.
So it becomes like this.
Yeah, so it becomes smaller
Okay.
and more cholesterol rich because
So
you've
then
got-
it goes into liver.
yeah.
Liver.
Yeah. So you've got all this lipid in there. Other things, carbohydrate can be being converted into lipid in the liver as well, and eventually all that mix will be packaged into very low dense lipoprotein, VLDL, and so that's how the liver is made. What the liver is then doing.
So actually, liver transfer this one into the.
Oh yeah, but it's not. What I was trying to say is that it's not direct. It's not just sort of A to B. It's A to C to D to E, different physiological stimuli, and then maybe F. You might have. You'll have some of that eventually working its way into VLDL, but it's not. It's not just input output.
Yeah. But during the process, is that because we still have some cholesterol inside this vesicle?
No. Well, no, that's been cleared in the liver. Yeah. So it's all been taken in the liver. In the liver cells, it's basically been degraded in the hepatocytes, in the lysosomes of the hepatocytes, and so that just basically chomps up all the protein, all the lipid, so it can all be sort of be reconstructed in eventually into VLDL, or make a fatty liver. Eventually, you can make a fatty liver as well. Yeah. So
the last step is you got a VLDL, but do you still have cholesterol or fat inside the VLDL?
Yeah. So from the liver. So this might be
Okay.
So this might be some cholesterol that you've actually got from the diet, but a lot of it will be the liver itself
So it's
is
the liver
synthesizing a
outside.
huge amount of cholesterol as well. Yeah, is that sort of make a bit more sense?
Yep.
Cool.
Thank you.
Okay, cool.
I just had it's not directly related.
Sure.
I was wondering, like, with, say, low carb diets, which everyone seems to be on these days, are they good or bad for your health?
Well, yeah, like the jury's out. So, okay. So, I guess the first question is, do they work? and there's been a few sort of increasing well-designed studies which have tested, and there's an inkling that they might create weight loss, but how much weight loss is due to the fact that you tend to self-restrict your calories because you can't eat too much protein and fat. Yeah, exactly. So that's one of the issues. If it's seriously ketogenic, which means making ketone bodies, which you can actually make from burning fat, that can be bad in the long run because, and they've done this with, from the 1920s, they've actually treated epilepsy with low carb diets, and they get a lot of kidney stones. And so the basically low pH of the ketone bodies in the bloodstream can strip calcium out of your bones, and you can then crystallise coagulation in your kidneys to make kidney stones.
So do they get lower bone density as well?
Yeah, I've actually seen that directly studied, but you can imagine that would be one consequence of that. The other thing is that certainly things like the Atkins diet, Atkins never worried about where the fat was coming from, what the type of fat was. And so, streaky bacon is as good as a bit of nice olive oil dressing or something like that on your salad. Yeah. And so there is certainly concerns that there would be an increased risk of heart disease because you're often eating more saturated fat. But obviously there are I'm seeing different low carb diets.
I mean, then again, like if you're consuming a lot of trans fat, but then your fat is really higher because you're not taking in as much carbohydrates, so you may be doing fat oxidation. It's less likely to be incorporated in certain.
Well, nobody really has known those which is a good question. The other thing is that there is certainly one of the downsides is that your brain prefers glucose. The brain in starvation it can use ketone bodies as energy, but it prefers glucose. And so there's been studies which have shown that people on low carb diets have more problems with concentration and all that. Presumably because they just haven't got any carbohydrates coming in to their brain.
But I mean, your blood glucose levels are going to be maintained by
to some extent. In starvation, it can still drop by about 20%. So it's working hard. It's working, like 20%, having a deficit of 20% of your starving is actually pretty good in terms of a homeostatic mechanism. That's not bad. But still, it's marginal cells which really convert into the carbohydrates such as where you probably and so I think that concentration that's been micrograms well studied. So there's jury's probably out. If you look at the on a diet, that's actually a high protein diet.